Okay, good afternoon and welcome to the DCRI Research Conference. It's good to see the conference so well attended, and I think it's a reflection of kind of the excitement and the buzz around biomarkers. And we thought that for National Biomarkers Month that we would have this compendium. Dr. Bohr is looking to see if that really exists or not. No, I just made it up. But there is a lot of excitement around biomarkers at the university level, certainly with the DTMI, the DTRI, and the DCRI all working together to raise biomarkers to its appropriate level. And as a kickoff, and I'll give John McHutchison an opportunity to talk a little bit about some of the new faculty members that are joining us in biomarkers. We thought that an RFA would be a good way to begin the process. So I'm going to spend a few moments going over the RFA that the DCRI has put out for biomarkers, and then we'll have some presentations for projects that are being submitted as a part of this RFA, and there'll be other presentations in the future as well. Let me first acknowledge Bob Harrington and John McHutchison, Kristin Newby, who are a part of this effort, and we certainly appreciate their support. So the purpose of the Biomarkers RFA is to stimulate discovery by providing an opportunity to DCRI faculty and to fellows to fund projects in biomarker science as an independent and emerging discipline. The DCRI has committed a total of $300,000 to this RFA. A minimum of 3 projects will be funded to a level of $100,000 each, but projects of smaller amounts would be considered as well. And we'll ask that the projects that are funded be completed within 1 year. Now, funding may include projects and statistical costs, but is not designed to support faculty salaries. The overarching goal is to stimulate both interest and growth in biomarker science within clinical trials at the DCRI. It's intended to facilitate the use of existing samples to obtain preliminary data that would then go on to support grant submissions, high-tier publications, and creating tangible examples of success stories that better position the DCRI and its faculty and fellows for future funded research. These are some of the minimal requirements for the first phase of funding. Category A faculty member or a Category B faculty member with an integral involvement of a Category A faculty sponsor. DCRI-based fellows with a DCRI faculty mentor may also respond to the RFA, and I've had probably at least a half a dozen or more fellows come and inquire, and I'm very pleased by that. Samples must have been collected through a DCRI clinical trial or project and be readily available for DCRI faculty use. So as you can see, what we're trying to do is to utilize samples that have been collected over time as a part of DCRI-related projects. And that's not to say we're dissuading people from collecting samples in future projects, but we want to make sure that the samples that have been collected are put to good use, and then in turn will support future research. Now, the applications must specify the laboratory to be used for the proposed analyses and provide assurance that capacity is available to initiate the project upon notification of funding. So in addition to having samples that have been collected as a part of a DCRI clinical trial or project, there has to be a laboratory that is designated to do the analytical work. And of course, there must be documentation from IRB approval for not only the sample repository, but if clinical data is going to be used as part of the analysis, of course, the IRB will need to be part of that. And also that IRB approval for funded projects using the repository will be required before funding is released. These are some of the timelines, so we're into it already. As many of you know, that we sent out an email, and that went to DCRI Category A and B faculty as well as fellows, in January just to determine the level of interest. And after about 25 or so responses, we thought, that's not bad for a first go-round, so there was clearly enough interest to pursue this further. Step 2 is an introductory biomarker science presentations, which is what we're doing today. And Deepak Vora will be one of our presenters, and Tom Urban will be another, and time allowing, I will be the third. And this will give you a little bit of a feel for how the projects are being designed. So there'll be 10-minute presentations and an opportunity to ask questions. The investigators are then asked to submit, and this is for all, not just for the people that are presenting today, a 2 to 3 page proposal that includes a background, aims, objectives, a DCRI project that is the source of the materials, the current site of storage, proposed analyses, and the laboratory where the work will be done. And the due date for that will be March 15th, 2010. So 2 to 3 pages. And then there'll be a review committee that is being chaired by John McHutchison. I will be part of that, Kristin, Newby, and Bob Harrington, to evaluate all the proposals that are received. And then from those, there will be 6 investigators who are being asked to submit a more formal proposal of approximately 5 pages that must include a detailed budget. The invitations to those individuals will go out April 1st, and then the due date for receipt of the formal proposals will be April 15th, 2010. So fairly tight timelines, but with brief, focused proposals, we thought that it was reasonable. The review committee will then notify finalists on the 22nd of April, and then finalists will present a brief overview of their work at the DCRI Research Conference on April 22nd, 2010. Following those presentations, there will be a selection from the committee, and actually the individuals who will be funded will be announced there and then. Monies will be awarded on May 1st, 2010, of course pending notification of IRB approval for the project. There are some expectations of course, and things that the awardees will be asked, and that is to provide quarterly progress reports to the Academic Review Selection Committee. And the reports will need to include progress to date, publications that may have come from that work, presentations, grant proposals submitted, and anticipated new research opportunities as a result of the funding. The funded investigators will also have an opportunity to present their finalized work within a year of when the award is given. So this is a brief overview of the RFA for DCRI biomarkers. I am very pleased, as is John and Kristin and Bob, with the level of enthusiasm. And as I look out into the audience, just seeing the number of people here and people shaking their heads, you're saying, well, this is a good thing. This is what we are needing as faculty members and as fellows. This will give us an opportunity to fund those future research efforts, and also to work closely with our colleagues in the DTRI and the DTMI. So we're all looking forward to it. I can stop and see if there are any questions before I have John come up and introduce some of the faculty members that will be joining us as a part of the biomarkers group. Okay, with that, John.
We should get on with the presentations, I suppose, but no, that was beautifully put together. Thank you. Rick's done all the hard work with Kristin, but it is very, very important. I think everybody sees it as an opportunity. We've got a world-class institution here. We're doing world-class important clinical trials. We're collecting samples, so we should be trying to explore response and toxicity, etc. We have some tangible examples of how we can do that, so easy to move forward. As part of the increased investment in biomarkers and biosignatures here, we have brought on 3 young faculty. We'll be supporting part of their time here to assist us and individual projects, an extension of the biomarkers work outside of cardiology, if you don't mind me saying that. Deepak, who's talking today, Vora, will join us as a faculty in biosignatures when he finishes his fellowship in July. Svati, are you here? Svati Shah is joining us in July when she gets back from maternity leave. We're not going to put her to work before then. Both of them are cardiologists, but they've got important ties to IGSP, CHG, Murdoch, and many other pharmacogenomic and other initiatives. So they are experts and will help us lead and help Kristin and Rick lead. And then also, who can't be here today, Kaya Patel, who's in our group, a gastroenterologist and hepatologist who has ties to Murdoch, IGSP and other initiatives. So all of these young faculty are keen, they live and breathe biomarkers and they want to do it, and I think us supporting them and having them involved operationally and in every other ways will drive biomarkers forward. So I look forward to hearing the presentations.
Oh, great. Thank you, John. And indeed, biomarkers know no boundaries, and we aim to follow that as a part of this process, that it will be balanced and it certainly will be highly integrated across all disciplines. So with that, Deepak Vora will come to the podium and share a few thoughts of the project that he has submitted, which is a pharmacogenomic analysis based on Plato. So we'll get this lined up for you.
All right. Hope everyone's having a good day. I'm going to briefly go through some ideas that we've been thinking about to explore in a recently completed clinical trial called Plato. And most of these are new and preliminary ideas, and so I welcome any input from the group here. And so, just as a means of overview, just briefly I'll talk about some of the background in terms of what's known about the genetics of CAD, coronary artery disease, and myocardial infarction, and try to highlight some of the unanswered questions out there, some potential hypotheses that we can explore, some preliminary data from some recent conferences as well as from our own group, and then how to approach this within a clinical trial context. And so I think it goes without saying that coronary artery disease and myocardial infarction are heritable traits. This is not something new, but that based on that known heritability, there have been multiple large-scale, independent, worldwide genetic association studies for coronary artery disease and MI, and the results of these large studies have really converged on one main genetic locus, and that's the short arm of chromosome 9. And this has really led to probably one of the most consistent genetic associations for a complex cardiovascular disease. And the risk allele is present in at least half of Caucasians. And as a part of this story, there's been a wide range of associations for related diseases, from AAAs to peripheral vascular disease to cerebrovascular disease, as well as some intermediate markers like coronary calcium and carotid intima media thickness. And so this is a recent meta-analysis that was performed of 12,000 cases and 28,000 controls, demonstrating that if you have one copy of this variant allele, I don't know how well you can see it, but this confers a risk of about 20%, 20 to 25%, and then if you have two copies of this risk allele, that risk increases to about 50%. And although the results of these genetic associations are robust, there's been some question as to whether or not the variants are predisposing more to the risk of coronary artery disease versus MI. And so I think we're all, at least in the cardiology community, are familiar with the different distinctions of atherosclerosis versus atherothrombosis. The patient who presents with stable 3-vessel coronary artery disease and a preserved ejection fraction versus the patient who comes in with an occluded thrombotic occlusion of a coronary vessel. And so, most of the genetic association studies that exist have used a CAD plus or minus MI case group and a non-CAD control group. And although this has been useful and has been very illuminating, it leads to some problems with confounding, because any case that has an MI in it would be confounded by the presence of underlying coronary artery disease. And so the existing study design that's been used for genetic association studies really don't allow you to separate the risk of CAD versus the risk of MI when it comes to genetic variants. And so to kind of put this kind of graphically, basically, I think this is the type of situation that we're in here in the top panel, where you have a strong genetic association at 9p21, and it seems to be associated with coronary artery disease, and whether you can separate the risk of CAD versus MI is not possible, I think, in the current type of study designs. And so whether or not they're more complex associations where you have the 9p21 variant independently associating with the risk of MI, or even the type of MI, is really kind of a big unanswered question right now. So there have been some attempts to try to dissect the disease process a little bit. This is a genetic sub-study from the group in Utah, and they have an angiographic biorepository that's very similar to our CathGen database. And to try to get at this answer of whether or not this variant is associated more with CAD versus MI, they kind of constructed 3 comparison groups. So the first is a group of patients with coronary artery disease, and they compared those that have an incident MI versus those that didn't have an incident MI. The second group are a group of patients who came into the cath lab with an MI versus those who came into the cath lab without an MI. And their final comparison group was a group of patients with coronary artery disease and MI and/or MI, and comparing them to a non-CAD control group. And so this is their results. So basically, if you look at the group that had incident events, or the people that had a prevalent MI, there's really no clear signal for an association with the 9p21 variant. If you remember from the meta-analysis, the point estimates should be somewhere around here, at least 1.2 for the risk per allele. And so, but if you looked at that third group of patients where they compared patients with CAD and/or MI to a non-CAD group, then you see the expected odds ratio of around 1.2 that was statistically significant. So suggesting that there's some separation in terms of what risk is being conferred by this genetic locus. And then finally, in another more recent publication, I'm sorry these curves don't reproduce as well as they should have, but this is a follow-up study of 2,000 patients with drug-eluting stents placed, looking for the incidence of death, MI, and repeat revascularization, and really finding no association with future events. So essentially, once you have a group of patients with coronary artery disease, knowledge of this variant doesn't inform prognosis or risk of future MI. So again, 9p21, probably the most robust association in the literature for CAD and MI. Some questions, though, that still remain in terms of which disease process is actually being conferred by this variant, and in terms of prognosis, whether or not this variant is useful after the diagnosis of MI is something that is up for question. So I'm going to shift gears a little bit in terms of mechanism. You know, 9p21 lives in a bit of a genetic desert. There's 3 genes that are downstream that are not known to be associated with the atherosclerotic disease process, and this is not uncommon for genome-wide association studies. But understanding the mechanism, or having some insight into the mechanism, is critical if we're going to devise strategies to mitigate the increased risk that's conferred by these alleles. There have been a variety of investigations looking at intermediate phenotypes to see what is the mechanism by which this variant confers its increased risk. And I think it's pretty clear that the 9p21 variant is not associated with an atherogenic or a thrombogenic profile. Initially, there was some thought that maybe the 9p21 variant was associated with diabetes, and that was confounding, or it was in the same pathway. But again, that has turned out to be not the case. A closer look at this region on chromosome 9 shows that there's 2 separate, distinct regions. One region that's associated with coronary artery disease, and one that's further downstream and not in linkage disequilibrium with the CAD block that confers the risk of type 2 diabetes. So these seem to be separate, and that's though adjacent to each other. And so I think we can say that 9p21 does not appear to be conferring its risk through a traditional cardiovascular risk factor. But the question is, are there more complex modes of genetic risk that could be associated with this variant? And so this is a paper that came out a couple of years ago looking at a group of patients with type 2 diabetes and then stratifying their risk not only by the 9p21 variant genotype, but also their level of glycemic control. And so what you can see here is that if you're in the lower two tertiles of glycemic control, that there is an increased risk of coronary artery disease as you go from 0 to 1 to 2 alleles. But then if you're in the top tertile, that this risk appears to be accentuated. And this was also a statistically significant interaction, suggesting that it's not only the presence of the allele or the presence of poor glycemic control, but the combination of the two somehow confer an increased risk. So suggesting that there's a gene by environment interaction that may be important with this locus. So again, in summary, a robust association with coronary artery disease. Unanswered questions include the distinction between CAD and MI, prognosis, and maybe how this genetic risk is being conferred. And so finally, as the last piece of background here, the rationale for having a molecular or genetic diagnosis for acute coronary syndromes. I think we're all aware that risk stratification is important after an MI. This helps us tailor the intensiveness of our secondary prevention strategies. And as we reach a point of limited resources due to cost and polypharmacy, we may want to identify patients who are at the highest risk after a myocardial infarction to tailor not only pharmacotherapy, but all of the therapies that we have to offer. And then there's this concept of a molecular diagnosis and whether or not knowledge of your genetic background, whether that can influence your behavior in some way. And that's something that, we've been exploring in some of our other studies, but whether or not if you know that you have a genetic variant and that may have predisposed you to have your event, maybe that will make you less likely to keep, or more likely to stop smoking, or more likely to keep up with your medicines. And that's a hypothesis that remains to be tested, but again, something that would provide the rationale for a genetic diagnosis at the time of MI. And then doing all of this within the context of a randomized controlled trial provides many other opportunities, such as being able to test novel therapeutics that are randomly allocated with respect to the genotype. And this, in the absence of knowing what the mechanism is for a known genetic variant, this would allow you to test novel therapeutics and maybe glean some insight into the mechanism of a genetic variant. So with all of this background, I just wanted to throw out 4 potential hypotheses that we've considered. The first is whether or not CAD or MI variants interact with cardiovascular risk factors to influence the risk of MI. Second, whether or not CAD or MI variants can influence the type of MI that you present with, so an ST segment elevation MI versus a non-ST segment elevation MI. And then going forward, looking if these genetic variants are associated with prognosis after an acute coronary syndrome. And then finally, if intensive dual antiplatelet therapy can provide greater protection of events versus moderate antiplatelet therapy in patients who carry a genetic variant. And so some recent observations to support some of these hypotheses. So this is a presentation from the AHA this year, and this is a sub-study of only 2,000 patients from the PROVE-IT TIMI 220 study. And this showed that the carriers of the 9p21 variant had an increased risk of subsequent event after their MI. They didn't have statistically significant effects, but the point estimate here was in the right direction. And this is slightly in contrast to the studies I've shown before. So this is suggesting that maybe there is some other independent risk of the 9p21 variant once you've accounted for their coronary artery disease, though this may have been underpowered. And this is some data that comes from our CathGen database that we've been working on here. And so we've genotyped the 9p21 variant in CathGen and stratified subsequent events, death, MI, and stroke by whether or not patients are reporting aspirin use. So obviously there's a lot that can be confounded in that piece of information, but I think it highlights an important aspect here, so that if you look at the 9p21 variant in these patients who are reporting aspirin use alone, you see a slight increase in risk that's not statistically significant, but that if you look at the group that's not reporting aspirin use, that there is now a higher statistically significant risk of subsequent events. And then if you combine these all into the same model, and test for the interaction for reported aspirin use with this genetic variant, the interaction between the two is significant, suggesting that there's at least some heterogeneity in the risk that's conferred by this genetic variant. It doesn't necessarily mean that putting people on more antiplatelet therapy will mitigate their risk, but that there's some difference in the risk that's conferred by this genetic variant and is hypothesis generating, I would say. So now, briefly, just to review Plato, recently completed clinical trials. And so this is a randomized trial of ticagrelor versus clopidogrel in patients who were presented with an acute coronary syndrome. All patients were treated with aspirin, and patients could have presented with either ST segment elevation or non-ST segment elevation at the time of presentation. Follow-up continued for 12 months after their index event, and the primary outcome was vascular death, myocardial infarction, or stroke. And as was demonstrated in this publication, that the assignment to ticagrelor showed a significant reduction in the primary outcome compared to clopidogrel. The trial overall was over 18,000 patients conducted in 43 countries. It was primarily a white population, 92%, with 38% presenting with an ST segment elevation MI. And so to conduct or to test the hypotheses that we've set out, we'll be taking advantage of an ongoing genetic substudy that's being conducted in Plato, where 10,000 patients with participants have DNA that's been banked, stored, and available for genotyping. And we consider genotyping the following genetic variants. I focus the talk today mainly on 9p21 because that's been the largest and strongest hit for MI, but since the initial publication there have been a variety of second-tier genetic variants that have been associated with MI that would be important to look at. And then in terms of the statistical approach, to look at gene by environment interactions, we would pursue a case-only study design to look for these gene-environment interactions. We could, to look at the association with the specific type of ACS, we could look for the prevalence of these alleles by MI subtype. In terms of looking at prognosis, we would be able to, since we have the clinical data at our hands, we'd be able to enter these clinical variables in a Cox proportional regression models to look for their influence on subsequent events and whether or not they interact with standard cardiovascular risk factors. And then finally, we'd be able to test for heterogeneity of treatment effect by looking for a gene by treatment interaction in the Cox proportional models. And we would want to adjust for a known determinant of clopidogrel response, the 2C19 variants. And there's also some interesting work that's come out of the KIF6 locus in terms of subsequent risk of MI after an acute coronary syndrome. And so this is just some power calculations and power simulations for the two types of study designs that I've presented. So the first would be for the case-only study design here on the left. And this is using some conservative assumptions, an alpha of 0.005, and this model is for a gene by sex interaction, and just showing that basically if you vary the minor allele frequency from 0.1 to 0.5, that we achieve a sufficient statistical power at an odds ratio of 1.1 to 1.2 for a gene by sex interaction, and you get similar results for a gene by treatment interaction. And so just suggest showing that with the power that we would have to be able to detect important outcomes in this genetic substudy. And finally, I've been focusing mainly on opportunities for DNA-based technologies, but there are plasma and proteomic-based platforms that are available through the Plato substudy. And so these are some of the additional opportunities that are also available. And with that, I'll end and have time for questions perhaps now or later.
Thanks. I want to really challenge us. I mean, if we're going to make an investment in biomarkers, it's Kevin Schulman. Not that there's anything wrong with what you just presented, but a couple of things. I mean, a 1.2 hazard ratio out of case control studies is meaningless. I mean, that's why why everything's bouncing around. But more importantly, it sounds like for 10 years people have been going down this one path about epidemiology, and cardiology's found nothing. Oncology's got tons of stuff. So what's going to be unique about what we're going to do? So genetic epidemiology and risk predictors, and even if you got 1.2, you wouldn't even do it in the clinic. So how are we going to decide what's clinically significant for us to go after? And then more importantly, given the platform that we have, what's going to be really unique that we could do here that Utah couldn't do or that Oxford can't do? Pharmacogenomics in a clinical trial seems like that's unique that other people couldn't have. But predicting MI or predicting CAD, I can do 10,000 epidemiologic studies and get that. What's going to be special about what DCRI is going to do that's going to either grow the science up to a level that's appropriate for clinical practice or have some unique insight into this and to make us different than all the people that have failed over the last 10 years?
That's a very tough question to ask a young guy that we've just brought on as faculty, Kevin. So why don't we save that question for later and save the other questions for later? But no, the point's well taken. But let's move on and hear Tom, and then we'll have some general discussion. And what you're raising are important issues.
So with that, I'd like to introduce Tom Urban. Tom is at the IGSP. He's done some very interesting work, and he's going to present some thoughts around the whole genome sequencing in Valproic-induced liver injury. So, Tom. Thanks.
Thank you. So I'll be talking partly on behalf of John McHutchison here at DCRI and my postdoc mentor David Goldstein at the Duke IGSP Center for Human Genome Variation. So the topic of the talk is drug-induced liver injury, and here I'm not talking about transient elevation of LFTs, but severe liver injury, frequently resulting in hospitalization, occasionally resulting in mortality or requiring liver transplantation. This kind of severe liver injury is the primary cause for drug removal from market after FDA approval, so there's a high not only a high cost to patients in terms of morbidity and mortality, but also a high economic cost to this. It's difficult to predict. It's called idiosyncratic. There aren't any clear clinical correlates to who might experience these kind of outcomes, and valproic acid in particular is one of the major precipitants of drug-induced liver injury, partly because it's so widely prescribed not only for epilepsy, but also affective disorders, migraines, a variety of neurological and psychiatric indications. So estimates as high as 1 in 5,000 prescriptions. This is probably on the high end of the incidence of valproic acid hepatotoxicity. It's actually even higher in some high-risk groups, pediatric patients, patients taking more than one anti-epileptic drug. And I'm hoping in the next few slides I'll be able to convince you that rare genetic variation has a potential to explain a lot of the risk for valproic acid-induced liver injury. I put this slide up here just to show that we're not entirely in the dark in terms of what causes DILI, what the biological mechanisms for DILI are. There are some known indicators, mitochondrial toxicity. There's thought to be immune response, both innate and adaptive. But what I'll be talking about from here on out is a more genome-wide approach to basically opening up the genome in a comprehensive way and asking where the genetic risk comes from, as opposed to trying to track down particular candidates or mechanisms that we suspect might be involved. And this is now possible due to large collaborative multi-center efforts to study drug-induced liver injury. In particular, the largest of its kind is the Drug-Induced Liver Injury Network, and actually the sort of nerve center of the Dylan network is right here at the DCRI, led by Jim Rochon, the PI of the Data Coordinating Center. And these 13 sites combined have the ability to cover 20 million patient lives. So even though these adverse events are so rare, we can still recruit something like 100 cases per year. And now the total is up, it's beyond 600 cases in the cohort that we have currently. So this allows, for the first time, some real genetic discovery. And so there have been a couple of studies that have shown definitive genetic risk factors for drug-induced liver injury. Two that have been discovered through genome-wide association studies, flucloxacillin and augmentin. Actually, the augmentin study was performed as a partnership between Dylan and another severe adverse event consortium, SAEC. These risk variants are common genetic variants that explain a little bit of the risk. So odds ratio is on the order of 2 to 3, which is nice, but because the events are so rare, the positive predictive value of the genetic test is not so high. They're not useful as clinical predictors. On the other hand, for valproic acid DILI, there's a recent study by Patrick Chinnery's group has shown that mutations in the mitochondrial DNA polymerase gamma, mutations in which are responsible for a syndrome called Alpers-Huttenlocher syndrome, have a greatly increased risk. So individuals that are heterozygous, so carriers of mutations that cause this disease, are at greatly increased risk. And this was a well-motivated candidate gene study because part of the syndrome is seizure disorder. And what happens is if you treat these kids with valproic acid, about a third of them end up with acute liver failure. So it's not surprising then to see that the carriers, the heterozygotes, are also at increased risk for this. And so recently, we at the Center for Human Genome Variation, in partnership with DCRI and Dylan, have performed a genome-wide association study of valproic acid-induced liver injury. And we found an additional rare genetic risk factor on chromosome 5 in the vicinity of two genes, thrombospondin 4, THBS4, and metaxin 3. These, I should mention, this is a rare variant, a variant with a population frequency of less than 1%. Typically, this type of variant would not be on a genome-wide association chip because they're intended to survey for common variants, those having population allele frequencies around 5% or greater. So it's kind of strange that this ended up on the GWAS array. But it's striking that, this rare variant was highly enriched in the valproic acid DILI cases. And the genes, although they might not have been at the top of the list of a candidate gene study, have some plausibility in terms of how they might increase risk for valproic acid toxicity. There's some suspicion that THBS4 is involved in innate immunity, and metaxin 3 is believed to be some essential protein for transmembrane transport of mitochondrial preproteins. So the idea that mitochondrial toxicity is part of the way that valproic acid induces liver injury sort of makes sense that this gene might come out of a GWAS study. So what I've presented so far is basically we have evidence that there are two regions of the genome that are harboring rare variants that confer a high degree of risk to valproic acid toxicity. Both of these were discovered basically by luck. So the polymerase gamma, you had a candidate gene that was such an obvious candidate gene that it didn't require, a very expensive study to make that discovery. The chromosome 5, the thrombospondin 4 and metaxin 3 associations, we're kind of lucky that the variants that were associated happened to be included on the GWAS array. Going forward, what we'd like to do is find additional genetic risk factors for valproic acid toxicity, but without the luck factor. So actually take a comprehensive view of the human genome and look for any type of variant, common, rare, single-site mutations, insertions, deletions, translocations, any kind of variant that could have some functional consequence that would predispose to VPA DILI. And the aim is to get something that actually has some use in the clinic. So, the idea would be to have some set of genetic tests that would allow you to screen out patients that basically should not receive valproic acid. And so the basic overview of the study is to sequence the entire genomes of some number of VPA DILI cases. The next step is to identify all of the variant sites by comparison to a reference genome or a set of reference genomes, and then screen out variants that have already been considered for association in the genome-wide association study, so basically all the common variants, and then prioritize them for a validation step. And the prioritization is based on some prediction of what each variant does, whether it changes the protein structure, causes protein truncation, affects splicing, any kind of large-scale inversion that might alter protein function. So that's the sort of the functional annotation. And then on the other side, look for enrichment in cases versus control genomes, either publicly available genomes or genomes that we have sequenced using the same platform here at the CHGV. And then once you have that list, then the next step would be to validate them in additional cases and large numbers of controls, because when we're looking at rare variants, we need to have extremely large cohorts of controls to accurately estimate the population frequency. And I'm not going to say too much about the technology, except that it's fully robust and operational in the Center for Human Genome Variation, the Genomics Core facility run by Kevin Chayana. And the capacity now is to sequence a genome in about a week. And that's at dense coverage, so at least 20-fold coverage. The idea is to sequence overlapping fragments of DNA across the genome so that you have high power to detect any variant that is there, and can also screen out sequencing errors, because there is some low error rate, and by sequencing to higher coverage, you can basically remove those things that are oddities and not real variants. So there are a couple of approaches to whole genome sequencing. So there's whole genome, where you're sequencing every mappable base in the human genome, and that currently costs about $10,000 per individual, and takes about a week, maybe 1 to 2 weeks, considering data processing and annotation. Alternatively, there are some people that are using targeted exome sequencing. So this is basically fragmenting the genome, enriching for just that 1% or so, or less than 1% of the genome that actually codes for protein, and just sequencing that. So that saves you a little bit of money, but there are some disadvantages there, because the capture step can introduce different types of bias, bias toward picking up sequences that look like the reference genome versus those that don't, bias toward known exons, so potentially missing protein coding exons that just haven't been annotated, variants that occur in non-coding regions that may be functionally important, and also structural variations. So these larger scale insertion, deletion, duplications, inversions of large stretches of DNA will essentially be missed by an exome capture. So that we're currently favoring whole genome sequencing for this. And this is just a screenshot to show you the bioinformatics tools for performing the annotation, and the biostatistical module are already in place. This is software developed by Dongnang Ge, also at the Center for Human Genome Variation, called Sequence Variant Analyzer. And what this does is it takes all of the identified variants and places them in their genomic context, so where they are in relation to coding exons, what they might do to protein structure, and also allows comparison to reference genomes, either publicly available genomes or in-house control genomes. And so again, this is the pipeline: sequence, annotate, prioritize, and then test for association in a kind of validation stage. And this slide is basically just to show that this has sort of already been done in-house. So Kevin Chayana, who has spearheaded a study of sequencing patients with hemophilia A who were multiply exposed to contaminated blood products, HIV-contaminated blood products in the early 80s, who never became infected, to try to look for basically protective factors from HIV infection. So we can ask in this sample of patients with hemophilia A, 10 cases, 10 controls. If we do nothing but ask what proteins in the genome are disrupted in the cases and not in the controls, we find that factor 8 is the top gene in that list, and factor 8 is the mutations in factor 8 are known to cause hemophilia A. But this shows that if we didn't know that, we would have found it using this strategy. So the conclusions are that this is using the most contemporary genetics techniques to address a problem of particular interest. It's the only initiative currently of its kind in drug-induced liver injury. It could lead to some clinically useful diagnostics, and it really takes advantage of the position of the DCRI in the Dylan network. And potentially, if successful, this could serve as a proof of concept that could be applied to other rare drug toxicities or common drug toxicities as well. And just want to thank Jim Rochon and Hans Tillman, Dylan and NIDDK, and the folks at the Center for Human Genome Variation that have got this sequencing pipeline up and running. Thanks.
Good. I think for the sake of time, what we're going to do is move forward. I'll have Deepak come up and join us. I want to make sure that we can get to some of the questions, and I think the best place to start is with Kevin's question, which is critical. I mean, in terms of complex genetic disorders, how do we use the information? How will that inform subsequent research? So, Deepak.
All right, I've had a little bit of time to think about this. Thank you, John. I agree. We're not going to be able to put together the types of databases that already exist for CAD and MI, like you mentioned. But I think the resources that we can leverage are the fact that we have outcomes data that within the context of a clinical trial, so highly refined and robust adjudicated data. We also have a randomized drug treatment, and I think that is another strength of a clinical trial that we need to leverage. And so the hypotheses that I outlined weren't necessarily prioritized based on what we have to offer through this type of database. But I think to answer your question specifically, if we were to focus on the things that we do best, it would be those things: outcomes and randomized drug treatment that we have to offer that others don't.
And Deepak, if I understood you correctly, and you didn't mention in your presentation, but you already have some data in terms of aspirin response with that particular gene mutation. Would you like to comment on that, just so that people see the link between the genetics and the treatment piece?
Right, so that piece of preliminary data, again, very preliminary, but just suggesting that, at least in our hands and on the patients that we take care of on a day-to-day basis that come through the Duke lab, that those that are reporting aspirin use seem to be at an increased risk for events if they carry this mutation compared to those who do report aspirin, who don't report aspirin use. And so that suggesting that antiplatelet therapy may mitigate the risk of this variant, and that's kind of the platform for taking this to a trial like Plato, where you have randomization to two different levels of antiplatelet therapy.
So I think Kevin's got a great question, and it seems to me though that do you cast your line for smaller fish, or do you cast your line for the big marlin, cardiovascular disease? So, I mean, if you go after something that's quite rare and specific, and, I'm obviously looking at the difference in the presentations, not biased towards Tom's, of course, because it's liver disease, but, going after a complex disease and going after a drug reaction or a specific response to a drug are completely different approaches. So I think we have to think of things differently.
Svati, do you have a comment?
So I think Deepak makes a lot of really great points. I just point out a couple more things. In relation to John's question about complex diseases versus sort of extreme sort of drug reactions, I think we have to be able to tackle both here at the DCRI. from the complex disease side, one of the strengths of working within clinical trials and these large databases at the DCRI that have been lacking in some of the previous studies is being able to define those we call them endophenotypes in genetics, but essentially being able to say, well, it's not all cardiovascular disease as a whole, it's not just MI as a whole, but really trying to understand what are the subgroups within those overall patients that we really want to understand. And having the exquisite databases that we have here at the DCRI really is going to be able to help us define those subgroups better. And the second thing I would say is Deepak has not presented some of the other wonderful work that he's been doing, where he's actually been looking at things beyond genetics, so looking at gene expression, looking at protein biomarkers, and looking at metabolic biomarkers. And that's really a strength that Duke has that we've been able to capitalize on, for example, in the Murdoch projects, that I think we're going to bring to hopefully bring to these clinical trials through the Biomarker Initiative.
I agree completely with you, and I'm not downsizing it. I'm saying we've got two distinct set of problems, and we have to have different approaches for different problems if we're going to, solve the biomarkers issue.
Yes. Absolutely.
Any questions for Tom?
Well,
You've got to have some liver disease questions.
No, I have a question. I've got one, thanks. But honestly, you made a pretty strong argument that even though the mutation is not a common mutation, it's not the rarest of the rare either. So if I understood you correctly, that there are enough people out there that have it where you would say you wouldn't want to necessarily use a particular drug if you carry a particular genotype because it puts you at risk. Am I understanding that correctly?
That's right. So I guess, as an example, the thrombosfondin 4 McHutchison 3 region, the population allele frequency is about 0.7%, so 7 out of 1,000. In the valproic acid cases, it's 25%. So that's a big difference, and that might be enough to make a difference as to whether or not you prescribe. Yeah. It's not fully penetrant. There are probably, most patients with that genotype can safely take valproic acid, but you might not want to risk it. Yes. In that case.
No, that's great, and because John is here, I must say that we are learning some things about cardiology and genotypes that may suggest we should use one drug over another. That's all I'm going to say. Please.
Manela Abdul Malik. I'm also with liver disease, and I really enjoyed your presentation. My comment relates to the ability to actually answer the question at hand, because by focusing on a very finite disease entity, an outcome, a rare outcome, you're going to be already looking at a group of patients that are highly characterized, and it may be difficult to know if the genotypic results that you get are associated with the disease at hand and why these patients are taking the drug that they're taking, or as opposed to the outcome that you're looking for. So my question pertains to the ability to do these biomarker studies beyond the aims presented, because ideally if you're going to do the study, it would be wonderful to use the Dylan network and the specimens, but even more ideal to have patients who are at risk for seizures who are taking valproic acid who didn't have the outcome in hand to assess whether the genetic characteristics you're characterizing define the disease entity that these patients have or are at risk for versus the outcome that they have that is a result of the intervention. In the standpoint from the cardiovascular databases, it seems like that might be there, but clearly you define two different studies with two different sets of resources and two patient characteristics. And are there abilities within the DCRI for crosstalk, if you will, across studies so that patients who are on valproic acid but may be participating in other trials or be in other data sets may serve as comparable controls for your Dylan network, because those are highly selected patients with a very unique outcome and the ideal control doesn't exist.
If I'm understanding you, you're asking, is there an opportunity for crosstalk and collaborations? I think the answer is yes, and we've seen it not just at the biomarker level, but at other levels as well. And your point is important. It's come up recently in some of the stroke trials, where in cardiology we have very, very large patient populations where stroke is one of our outcome measures, but the trials are typically not powered to look specifically at that endpoint. However, we do have lots of patients where that crosstalk with our neurology colleagues and neurosciences colleagues could certainly bear fruit, and it's something that the biomarkers program is certainly trying to emphasize. So, yes.
Well, I think just in response to the general point that the severe adverse event might be completely confounded with the indication for it, that's something that we definitely have to consider. So, so definitely making partnerships with, we have existing partnerships with the Department of Neurology and Psychiatry that can help us sort out those kinds of questions. But, of course, the more crosstalk and collaboration, the better, especially here where you have so many well-characterized patients.
But if you have 1,000 patients as your controls from the general population, where you know the human genome, are you comfortable that that variant is associated with it or not?
It depends on the allele frequencies and where those 1,000 people came from, how well they're phenotyped. But yeah.
I think we have time for one more comment or question, and then people can come up and ask Deepak and Tom questions. So, Pam, do you want to ask the last question?
Yes, as an imaging biomarker person, where we've been using that kind of testing, imaging testing, to identify disease characteristics or drug response characteristics for many years, we found that we need to get beyond, or at least the next step, once we have the technical performance of a test, which is partly where you guys are right now. We're understanding your assays and understanding what the sensitivity and specificity are for a particular gene mutation in a disease or a toxicity. The next step is to understand how that affects your diagnostic thinking and therapeutic thinking about patients. And then ultimately, do you change care based on that test? And so one of the things that we can do at DCRI that would be very different from what most people are doing is to start incorporating diagnostic testing as randomization points in protocols. And many of you know that we just launched the first one of that in cardiovascular imaging at DCRI this year, PROMISE study, where we actually say, do we do this test or that test, and how does that affect outcomes, costs, patient quality of life? And ultimately, if the genomic or genetic tests are going to be part of reimbursed routine clinical care, they will have to come to that bar where they improve outcomes in patients. And we have a superb outcomes group here, with superb trials group here, where we can get to that level of proof of principle, I think, faster than anybody else in the country.
Good. So, John, would you like to close with a few comments?
No, thank you, guys. Great examples. Thank you, Rick. I did forget to say one thing this morning, that is, as well as the investment in the young faculty, and if you've got projects that you're thinking about biomarkers, you now have the young faculty, and you've met the young faculty who you can approach, along with Diane Cheeseborough and her operational team. But I forgot to announce that along with this sort of initiative and projects, we're going to have a fellowship in biosignatures as well. So the DCRI will invest in somebody who will have a 1 or 2 year fellowship that will be related to the faculty efforts and developing biosignatures internally.
Good. Well, thank you, John, and thank you both, Deepak, and for being here with us, and thank you all for joining us as well. Thank you.